Hello friends, today I'm going to do some more drawing for you. Not really drawing, this is going to be a table, which isn't as exciting as doing a drawing, but there's a lot of information that I would like for you to know about the components of the Escherichia coli replisome, which is to say the DNA replication machinery that enables the chromosome of E. coli to be copied. Now, why specifically E. coli? Because this is the one we know the most about, is one reason. Your results may vary, as we will see in a future video. Just an example of how things may vary. But for the purposes of this video, it's all just going to be all E. coli all the time. Okay? So, I'm going to use my handy dandy straight edge here. And we're going to have the name of the component and the function of that component. Using my fancy 4-color pen today. All right. So first component I want to tell you about is a protein called DNA, which requires energy to function. So it's often, when it's functioning, called DNA plus ATP. Notice it's big A, big D, little NA. That tells you that it's not the nucleic acid itself, but it's a protein interacting with the DNA, and you'll see that with some of these other components that will have not surprising names like DNA B, C, D, etc. But DNA A binds the origin site, which hopefully you recall, that's a sequence of nucleotides with some simple sequence repeats called ORI-C. So this is considered to be the initiator protein. So the ORI site has 12 repeats of a nonomer, or a 9 nucleotide sequence, plus some other sequences that signal where the beginning of replication should begin. Okay, so the second component is going to be DNA B. DNA B, this is our helicase. And helicase is the enzyme that unwinds the double helix, enabling the DNA polymerases to gain access and read off the template strand for adding complementary nucleotides. And helicase is a hexamer that's kind of like a loop. It forms a circle around the template strand. And because it has to be assembled around the template strand, it requires another enzyme, which is DNA-C. And DNA-C is the helicase loader, which assembles the 6-part helicase molecule around that template strand. Try to draw that 6 helicase loader. There we go. Okay. So next component, you might think it's going to be DNA D. It's not. It's going to be these proteins that are not really enzymes, but they're just called SSBs, for single strand binding proteins. And these are necessary because, as you recall, the double helix is a very well assembled molecule which has the reverse complementary anti-parallel sugar phosphate backbones held together by phosphodiester bonds. And it really wants to simultaneously snap back together unless there is something actually physically holding it open. So we need a couple different enzymes to make sure that we can get access to the nitrogenous bases, hold those hydrogen bonds apart long enough that we can do the replication. You know, you can kind of think of it like, you know, when you go to the dentist, you know, they'll be like, open your mouth, open, a little wider, a little narrower. Sometimes they have to put a block in there to make sure you don't close your mouth when you're not supposed to, because the natural position for the mouth is to be closed. So sometimes we need different things to make sure that we keep that thing that's open. Or like when you pull a spring apart, or if you have like a rope and you pull the strands of the rope apart, it's been in a rope conformation for so long that you pull the strands apart, you let go, and it immediately snaps back to being rope shaped rather than stranded. Okay. So we're going to jump up to DNA G. What happened to DEF? I don't know. And you don't need to know either. So never you mind what those things are. But we do have a DNA G, which is our RNA primase. Now, remember that with our DNA polymerases, the molecules that actually add deoxyribonucleotides to the growing DNA strand, they only work under very stringent conditions. They have to have a free 3' hydroxyl on a tiny section of double-stranded DNA in order to function. So unless there's at least a little bit of a double-stranded molecule, then DNA polymerases don't know what to do. So RNA primers are added, just a few nucleotides, maybe 10 nucleotides of RNA. RNA is like, I don't care, add me to anything. I don't care if it's complementary, I'll be a part of it. So RNA is added at least temporarily, and sometimes rather sloppily, like the RNA nucleotides aren't always, completely complementary to the sections of DNA that they are added to, but it's okay because they're temporary. They're going to be pulled out later by enzymes that I will tell you about. So this brings us to, trying to keep you consistent here, DNA polymerase 3. DNA polymerase 3 because it was the third one characterized. And the DNA polymerase 3 is what adds deoxyribonucleotide triphosphates or to the primer. Well, it's actually ultimately going to be adding nucleotide monophosphates to form that consistent sugar phosphate backbone. But the building blocks, the nucleotides as they are built in to the growing copied strand of DNA, start off as nucleotide triphosphates. So deoxy ATP, deoxy GTP, deoxy CTP, deoxy TTP. And it is the cleaving of the pyrophosphate, those two phosphate groups that are on those nucleotides that provide the energy to make that very, very strong bond that is the phosphodiester sugar phosphate backbone. Okay, but DNA polymerase 3, I'm going to add in I'll use a different colored line, or maybe I'll just use a blue line. I'll use it to kind of indicate that the holoenzyme contains multiple subunits in and of itself. So we've got a structure called a beta clamp. I'm moving from the names into the structure section over here. That's okay. It's could be like a little, like kind of an appendix in a sense. So the beta clamp is going to clamp single-stranded DNA. So it follows behind helicase. So we need this additional clamp. So clamps have to be added on before we can start adding the nucleotides. And it's the DNA polymerase binds to the clamp, so it knows where it's supposed to be feeling around for those free 3' hydroxyls. We've got units alpha and epsilon and theta, alpha, epsilon, and theta, which make up what we call the core enzyme, the one that's really doing all of the work of breaking off those pyrophosphates. And then we've got tau subunits. That's a Greek letter tau, which are called the clamp loader. And the clamp loader is what actually puts the clamp, the beta clamp, onto the single-stranded DNA. And this can be done once in the leading strand, but in the lagging strand, you have to continually add new clamps to recognize the primer sites, the primed sites, to add those nucleotides to the free 3' hydroxyls. So the clamp loader, it kind of looks like an octopus. It's like kind of like feeling around on the DNA molecule and adding a beta clamp where it is needed. Okay, so that's all of the components of that DNA polymerase 3 holoenzyme, the whole being made up of many parts. And that is going to bring us to DNA polymerase one, and DNA polymerase 1 is going to remove the RNA nucleotides of the primer, and it replaces them with DNA nucleotides. So it recognizes on the end of the previous Okazaki fragment, for example, that there's a free DNA 3' hydroxyl that's attached to an RNA molecule that's bounded by an RNA molecule, and it recognizes that those RNA nucleotides, they're just a placeholder until DNA nucleotides can be added. Okay, so let's see what color I'm on here. Boop. And I'm just going to also add in here that this is often abbreviated as DNA pol 3, and this is DNA pol 1. Why are they numbered like that? Even though they don't act in that order, it's the order of their characterization, their discovery, and kind of the identification of their function. Okay, so next component of our replisome is going to be DNA ligase. And ligase fixes the what's known as NICs in the sugar phosphate backbone. So it's going to end up joining the Okazaki fragments together. So the issue is that with DNA polymerase 1, remember DNA polymerase is very stringent in their activity, so they can only add nucleotides to a free 3' hydroxyl. So that's part of the reason why we need to have the priming, and you need to have the Okazaki fragments because you've got the leading and the lagging strands. But when we remove those RNA nucleotides using DNA polymerase 1, then there's going to be one bond in the sugar phosphate backbone that the DNA polymerase 1 cannot heal over. It's at the end of where the primer was added. It's the very last connection in the backbone. But we don't want that. We want the DNA to be completely intact. So we need a whole other enzyme called DNA ligase, which fixes those last bonds that are not replaceable by DNA pol 1 in the sugar phosphate backbone. Okay, so just a couple more things I want to add to our replisome. One is a substance called TUS, and this is going to bind to the terminator sequences, or the TER sites, at termination. So as we're getting ready to terminate our replisome, because we've got a complete copy of the chromosome, we're going to use this stuff called termination utilization substance, or TUS, which is going to bind. And essentially, it's like a, okay, let's not go and just copy this thing all over again. Let's just copy it the one time and be done with it. So we need TUS. And then the last thing is, I spoke before about type 1 and 2 topoisomerases. Well, it turns out there's a couple more topoisomerases that specifically act at the end of replication. To separate the daughter chromosomes. So we have 2 copies of the DNA from the mother chromosome, and sometimes they can be twisted up in a couple different ways. I'll describe it in another video, and they require special topoisomerases to make sure that each daughter cell gets 1 copy, and only 1 copy, of the replicon, the copied DNA. Okie doke, that's it.